Welcome back, folks, to Objective 4, Part 4 of our unit on the nervous system. Today, we are going to hit the brain, or why do you have a bowl of oatmeal on your neck? I say that because the brain is very soft, almost the consistency of congealed oatmeal in some cases, so it's not the nicest looking thing in the world, but it does a lot for us. So here we're just going to be kind of going through the different parts of the brain and their basic functions. We're not going super deep at this point. If you take a neurophysiology or neuroanatomy class, or you're in psych, you might go a little bit deeper. We're just going to give an overview on this in this section of notes. So, development starts out when you are young as a neural tube in embryological development and fetal development. Here we can see it about 5 weeks as an embryo. The different parts here of it, very, very basic. Okay. Right here, as you get a little older at 13 weeks, starts to look a little bit more like what we've got for a brain here. We got the cerebral hemispheres in brown, the diencephalon, the brain stem, and the spinal cord coming down. You might liken this to like a bird brain or reptile brain. Pretty small as far as the relative sizes of things. Down here, we've got one that's more like a mammal brain, like a dog or a pig or cat or something like that. Looks a little bit more like ours. And then up here, we got ours. And notice how much larger the cerebral hemispheres are relative to the other parts of the brain. This is what gives us our brain power. It kind of sets us apart from most other organisms on the planet. The brain has some ventricles or some empty spaces inside of it. Not empty, they're filled with cerebral spinal fluid, but these empty spaces called ventricles, you can see where they are in here, the main ones anyway. They kind of act like support for the brain because you don't have like tendons and ligaments holding your brain up and supporting it inside your skull, holding it to the skull. It's just kind of floating in cerebral spinal fluid up there. And it's floating on itself because there's cerebral spinal fluid inside. Like I said, the brain doesn't have an extremely strong structure to it. It's pretty, I mean, you saw a brain, you could poke your finger right into it pretty easy. You've got rid of the skull and things like that. So it needs quite a bit of protection. So we got the skull around it, but it's also why it's bathed in cerebral spinal fluid. It's basically like if you were driving down the road in a giant pool in the back of a truck or something like that. If it crashed, you wouldn't feel, you'd slosh around a bit, but you wouldn't get smushed. The ventricles inside provided some buoyancy on the inside, so it kind of floating on itself, kind of like a big old whale in the water. It's fully surrounded by water and supported, and it's good to go. You bring it up on land, and it can crush itself under its own weight. Same kind of thing with the brain, and that's why we have ventricles in there. 4 major regions that we're going to tackle, and then we'll go into sub parts. But the cerebral hemispheres, the brown stuff we see on top here, the diencephalon, what's in purple here, kind of in the middle, the brain stem in green, and then this darker brown pinky thing back here called the cerebellum. It's not an umbrella, cerebellum hanging off the backside. We're going to start out with the cerebral hemispheres. Forms the upper part of the brain and makes up about 83% of its mass. So you see here, diencephalon, cerebellum, and brainstem. Here are the cerebral hemispheres. 83% of our brain by mass, not all brains. This is what kind of sets us apart, the percentage of our brain that is cerebral hemisphere. It's got ridges, okay? These little bumps are called gyri, and the grooves are called sulci. And the deep grooves between various areas are called fissures. And then you got one big longitudinal fissure. This brain's kind of split down the middle already, but there's a big fissure here between the left and right hemispheres, the two halves. And we got 3 basic regions. We got the cortex, or gray matter on the outside, white matter kind of in the middle, and then basal nuclei, which are gray matter scattered within the white matter. Okay. Major lobes, Gyri, Sulci, and the cerebral hemispheres of our brain. Very deep sulci divide our hemispheres into lobes, structurally into lobes. Each of those lobes kind of specializes in different things to a certain extent. We got the frontal, parietal, temporal, and occipital lobes. The parietal lobes, back here, that's our main somatic sensory area. where we recognize cold, pain, touch, and those types of things. If an area back here has, okay, we have in that parietal lobe, the very front of it is the somatic sensory area right here in blue. If that part of your body has more sense receptors, you're going to have more space in the brain. So you actually have more space in your brain for your hand in the somatic sensory area, dedicated to your hand, than you will to, say, your entire back. Even the back is way bigger because you have so much more, so many more nerve endings in your hand, so much more sensitivity there. You need more processing power to deal with all that information coming in, so you need more space. The sensory pathways are crossed left to right. It's called contralateral, or opposite sides, contralateral. So you may know that the left side of my brain here controls my right hand. The right side of my brain controls my left hand, or feels for my left hand. So we can see here is the somatosensory area right there, and this is the parietal lobe here. Here we can see a nice diagram of the primary sensory cortex, and it kind of got mapped out on its surface where it's getting information from. So we can see, look at all this space dedicated to the face and the lips, relative to, say, it's way less than this for the whole body here. Okay, and then you got quite a lot of space for the hand. Even got a decent bit of space for teeth, tongues, gums, pharynx, because you got to know what you're eating and be able to swallow it, coordinate it. And then you got intra-abdominal stuff down here. So these are the areas, and you can see what I was talking about. The more sensation an area has, the more brain space it needs. All right, let's move on to the occipital and temporal lobes. Very briefly, occipital lobe in the back deals mainly with vision. And that's why you'll see when we do a brain dissection later, you take a look at the eyes, okay, and the big, thick optic nerves going back to the occipital lobe, you'll be able to follow those because they're pretty thick, carrying a lot of information, but they have to go all the way back to the occipital lobe, and they cross because it's contralateral again, right? Left eye is right over here and like this, so opposite sides, and they cross, and that cross is called the optic chiasma. The temporal over here, that and kind of underneath, kind of on the sides and wraps underneath, that deals mainly with auditory and olfactory areas. Okay, so those senses are kind of controlled here. The cerebral cortex, that's the very surface part, the gray matter. I showed you that in this thing before. The darker areas on the outside, the gray matter, that is the cortex. Counts for about 40% of the mass of the brain. So about half of the hemispheres. That's what enables sensation, communication, memory, understanding, and voluntary movements. Most of the stuff we think about the brain doing for us. Each hemisphere, like I said before, acts contralaterally. They control opposite sides of the body. They are not generally equal in function. There is some specialization between the left and the right hemispheres. It's not total, but there's definitely some specialization. And no functional area in the brain acts alone. In order to get conscious behavior, it's going to involve the entire cortex, signals going all over the place to different areas of the brain to kind of process everything. As far as hemisphere dominance goes, your left hemisphere controls language, logic, and math type stuff. Tends to be a little bit more on that end of the spectrum. So if you're a left-brained person, you're more into logic, math, language, logic, language, left. Okay. The right hemisphere, a little bit more about visuospatial skills, emotion, and artistic skills. Like I said, they don't solely reside there, but it tends to lean that way. All right, the primary motor cortex, okay, is in the frontal lobe. So here is our sensory cortex here in the blue, and in this kind of pinkish whatever color here, the backside of the frontal lobe, that is our primary motor cortex. And that's involved in the conscious control, conscious, not involuntary stuff, but conscious control of precise, skilled, voluntary movements. So it's what's going to send out the information to reach out and grab the glass of water and take a drink, or move the tongue or the lips or whatever to speak. That's happening there. And once again, the majority of neurons deal with areas of fine motor control. So you're going to have a lot more brain space dedicated to the areas that have a lot more motor units or fine motor control, right? So I see it here in this diagram right here. And then we can see the control areas here. Lots more space to the hand than, say, the whole trunk and all that stuff here. Broca's area bears mentioning. It's present in one hemisphere, usually, but not always, the left hemisphere, because we have language over there, and it deals with, it's a motor speech area that directs the muscles of the tongue. So we have, it's one of the areas you can tell what part of the brain you might have had a stroke in, if it affects your ability to move your tongue to process and produce words. Maybe the stroke may have been close to your Broca's area. Ooh. And it's active as one pairs to speak. So we can see this with brain imaging, where we can measure the amount of glucose used in different parts of the brains. We can see that that area becomes activated when you speak. So the prefrontal cortex, so the front of the frontal cortex, okay, this part right here, okay, is involved with intellect, cognition, recall, and personality. It's necessary for judgment, reasoning, persistence, and conscious. This is kind of where your personality, this is where you reside in that part of your brain. Okay. This is the part, if they did a lobotomy in the past, they rarely do them now, where they basically scramble or mess up this part of the brain. That is lobotomy, and that would basically get rid of the person's personality. This is what was done to the Kennedy sister who was up at St. Coletta's here in Jefferson County a while ago, and why she was there. All right, the cerebral white matter. These are fibers that are deep in the brain, closer to the middle, and they're myelinated. So, like I said before, they're responsible for communication between the different parts of the cerebral cortex and the lower central nervous system, the spine, and areas of the areas of the cerebrum. So basically, they are high-speed transmission lines to allow the different parts of the brain and central nervous system to talk to each other. Here we can see kind of some of the fiber tracks, the wiring. We got wiring going between different sections here. We got long distance communication between the front and the back. The green ones are going side to side, connecting the two lobes of the two hemispheres of the brain. This is called the corpus callosum here. Sometimes you may hear about somebody having their corpus callosum cut. So this corpus callosum may be cut in cases where people have severe epilepsy, which is basically an electrical storm going on in the brain, spreading all throughout the brain. And they found, if they cut that and sever the two halves of the brain from each other so they can't communicate, it kind of stops those epileptic seizures from progressing. And you get some very interesting things. If someone wants to do some research on people who have their corpus callosum cut, it's some very interesting things. I might show some videos of it in class one day if I can find some online. What I do have for it is on old VHS tape, but I can't play that for you guys right now. So I'm going to see if I can find some that illustrate some of the interesting things that happen when a person doesn't have their corpus callosum and how their brain works differently. Here we can see a frontal view. We can see corpus callosum here, and we can see the fiber tracks from this side of the brain crossing over down here in the brainstem to the other side. This is why they have contralateral control, because the fibers cross right there. Why that is the case, I haven't read any or heard any I don't want to say well documented, but a widely accepted hypothesis on why we have contralateral control, why it crosses over like that. It's not just your left side controls your left hand, your right side controls your right hand. Something to do with our development in some very ancient ancestors in the animal kingdom. Basal nuclei, I mentioned them briefly before. They're areas of gray matter deep within the white matter. Kind of regulate voluntary motor activity. I almost like to think of them like old-fashioned switchboards. When you used to have operators in the phone lines way back when, and you get a call in and they connect this caller to that caller with a little plug-in wire, and they literally connect them and decide where that call was going. It's kind of what the basal nuclei do for motor activity. Kind of make those connections, shunt the signal off to the right place. You can see some of them in here in this image.
We began our journey into the human brain here on the campus of Dartmouth College in New Hampshire. I'd come to meet one of the world's leading brain scientists, Mike Gazzaniga, and a man he's worked with for over a decade. A man with two brains. You've been working a long time with Dr. Gazzaniga?
14 or 15 years.
It doesn't seem like that long, does it?
Well, the collaboration began when Joe had surgery. And you had this procedure to correct an epileptic problem,
yes.
Is that right?
Trying to stop the seizures. I was having seizures like every day or so, or sometimes 2 or 3 a day.
To control Joe's epileptic seizures, a surgeon severed the connection between the two halves of his brain. Cutting the corpus callosum like this prevented the spread of the electric storms that caused his seizures. But it also prevented the left and right halves of his brain from communicating with each other. In the years since the operation, Joe's epilepsy has been under control. He now earns a living at an egg farm, and in his everyday life, he's largely unaffected by the fact that his left and his right brains work independently. Do you feel any different when you think about something than you did, any differently from the way you felt before the procedure?
It's just got a backup brain, that's all.
That's something everybody could use, right? I found out how true that was right away when I was asked to draw a different shape with each hand. In a brain like mine, roughly speaking normal, at least all in one piece, the left half of my brain controls the right side of my body, while the right brain controls the left side.
Oh, no.
But because the two halves are connected,
Nothing wrong with that.
getting each hand to work independently isn't easy.
Well, we're seeing the fact that Alan's hemispheres are connected, and that the motor messages from one are confusing the motor messages in the other.
I was just drawing an upside-down duck.
Okay.
But when Joe is given the same task, his two hands operate as if controlled by two separate brains. What's happening is that each half of Joe's brain is given a separate instruction. He's asked to fix his eyes on the cross in the center of the screen. Anything flashed to the right of the cross goes only to his left hemisphere. Things to the left go to his right hemisphere. Because the two don't communicate, each hand does only what its half of the brain sees.
Wow, look at that. It's really like two different people doing the same.
That's right.
Same task.
That's the idea.
Okay, Joe, I want you to keep your hands out in front
In
of
an
you.
experiment that's now a classic in brain research, Mike Gazzaniga over 30 years ago used a similar setup to find out if the two halves of the brain are specialized to do different things.
Ship.
Joe is being flashed a word only to one half of his brain. Words flashed to the right
Storm.
are seen only by his left brain, and Joe can report seeing those words just fine.
Piano.
Good.
But when a word is flashed to his right brain,
Didn't see that.
Okay. Joe, I'm going to ask you
but now watch what happens.
to draw that with your left hand.
You get me lost.
Why don't you try drawing another picture of it over here if that will help you.
Oh, phone.
Oh, okay.
Grr.
It's almost as though somebody has given him a secret communication.
That's
and
right.
Now he knows that it's a telephone. But up until then, he was blind to it.
Exactly.
When Gazzaniga first did this experiment, it instantly proved that the ability to speak resides almost exclusively in the left hemisphere. Not until he sees what his right brain is drawing is Joe able to name it.
He said church, didn't he?
After looking at the picture.
But he had to figure it out about as long as we did. That's really interesting. It's a picture here of somebody communicating almost with another person.
And the communication is not occurring inside the head, it's occurring out on the piece of paper.
Yeah.
Blob. I really don't know.
You want to draw a little bit more?
So far, Joe has been seeing only one word.
Cat. That's a cat
Things get
sound.
even stranger when he's flashed two words, each to only one half of his brain.
The right hemisphere saw toad.
Yeah.
And so his left hand draws a toad.
So there's the toad.
Oh, it's a toad.
And this time, I was able to guess what was coming.
Will they put a little three-legged stool in there later, or what?
Joe's speaking left brain saw a stool. Saying the word lets the hand that's controlled by his right brain in on the secret.
That's great. That's really interesting. And if he had seen that with the corpus callosum intact, he would have drawn a toadstool.
Right.
Not a toad and a stool.
Right, exactly the point. I've been doing this for 35 years. Yeah.
And
it gets me every time.
Yeah, it must. It must.
This time, instead of naming the word, I want you to point to the word.
Again, Joe sees two words simultaneously. Bell goes to his non-speaking right brain. Music to his speaking left brain. When asked to point to a picture of what he saw, he chooses Bell. But when asked why...
Okay, why'd you pick that one?
Music.
Music?
And when asked to explain...
It was music and bell, and it was a few minutes ago, last time I heard any music was coming from the bells out here.
Uh-huh.
Banging away.
So the bell's outside here?
So, but good enough answer to me.
What's extraordinary is that Joe's speaking left brain concocts a plausible story of why he pointed to Bell, even when some of the other pictures more obviously represent music. Gazzaniga believes this determination to find cause and effect, this desire to explain, is the left hemisphere's most marvelous property.
One of the unique things to the human brain is this need to interpret why two events occurred. What was the antecedent to this? What caused this? And if you can imagine that a species like us that has that little chip in its brain that asks those questions is going to survive rather well, because it's going to figure out more about the nature of the world than a species that doesn't have that.
Okay, Joe, I want you to keep your hand...
But as I was about to discover, the right brain has a very useful survival skill all its own.
What do you think will happen here?
So for you, we're doing a live experiment. Never done it before.
The experiment involves the 16th century Italian painter Arcimboldo, who made faces out of fruit, flowers, meat, even books. From other research, there's reason to believe that the ability to recognize faces is located exclusively in the right hemisphere. So Mike wondered if Arcimboldo's paintings would look different to each of Joe's two brains.
So will his left hemisphere say, I saw a potato, I didn't see a face? And will his right hemisphere say, I saw the face, and not comment on the fact it was made out of a potato?
You're going to see a figure followed by a choice of two words.
If this works, it'll be terrific, but we'll see. So here it is, live.
Okay?
The first painting goes to the right hemisphere. And Joe points to face. The next painting goes to his left brain. And this time he points to fruits. Mike seemed pleased.
Are you having a moment?
This is too good.
Again to the right brain, and Joe sees it as a face.
Point it down.
But to the left brain,
Face made out of books.
Point it up.
Pointed to books.
Are you happy with what you're
Fruit.
doing?
It's unbelievable. He's doing it. You see that?
It's good. It's going so fast. It's shifting from left to right so fast. I can't keep up with you. You're used to looking at this.
When you show him a face in the right side, the left hemisphere,
Yeah.
he's focusing in on the elements that made up the face.
Yeah.
When you show him the exact same picture in the left field, going to the right hemisphere, he's focusing on the face and not the elements.
And not the elements. If you came down from another planet and you saw faces and vegetables, you might not think there was much of a difference among them. But the brain seems to be made up in a certain way to say faces are very different from other objects.
That's right.
And one side of the brain specializes in faces.
Exactly right. Exactly right. It is an adaptation that we have to detect upright faces. It's a very important. You can imagine in an evolutionary time that all of a sudden you have the ability to detect quickly an upright face. You want to read the expression on that face. You want to know if it's friend or foe. You want to have a set of questions about that face.
the right brain might be skilled at recognizing faces. But when it comes to what gives the human mind its power, the ability to reason, to invent, to interpret the world around it, Mike Gazzaniga's 30 years of research has taught him which hemisphere he wouldn't want to be without.
The old phrase around our lab is, don't leave home without your left hemisphere. That's where the action is.